Unlock the future of pathology with Pathology News, your ultimate source for cutting-edge insights, expert analysis, and the latest breakthroughs in digital and computational pathology. Don't just stay informed, stay ahead.
Hello and welcome to Pathology News. Today's a really exciting day because we are joined by Eric Walk, who's the Chief Medical Officer over at PathAI. And in this podcast today, we're going to be able to pick Eric's brain on AI, where it's going, and how we're using it in clinical diagnostics and in a research capacity as well. Eric, welcome to Pathology News.
Thanks so much, Sid. Thanks for the invitation and also the interest in the field of digital and AI pathology. I'm really thrilled to be here.
Yeah, no, it's really exciting to have you here, and I can't wait to get into this discussion. Now, Eric, as a new member or a new guest on our podcast, I was kind of hoping that you could give us a bit of an introduction to yourself, perhaps go into your background and what drew you to this area.
Yeah, sure. Happy to. So my background is as a pathologist. A pathologist, as many people know, that's the type of physician that traditionally looks at glass slides, makes a diagnosis of cancer and other diseases. My career as a pathologist has been non-traditional and I think very interesting because I practiced pathology just for 2 years, and then I entered the field of precision medicine and pathology innovation, as I call it. So initially, I had a job at Novartis Oncology, where I had the privilege of really seeing the birth of precision medicine, as we call it today, working on targeted therapies and biomarkers. I then moved to Tucson, Arizona, where I live now, and worked as the chief medical officer for Ventana Roche Tissue Diagnostics, where I continued my work on precision medicine and biomarker development using technologies like immunohistochemistry. And then in 2021, I was really interested in this relatively new field of AI pathology and joined PathAI as their chief medical officer. And that's my role today, where I oversee for the company several functions, including quality, regulatory, clinical, medical affairs, and our pathology team. So it's been an incredible journey as a pathologist. And as I said, the focus of my career, the theme has been innovation in precision medicine.
Yeah, great. Thank you so much for that, Eric. You've got a really interesting background. I think PathAI are very pleased to have you there. We're grateful to have you. Now, I'd like to start today's conversation a little bit differently to how we usually do things and, jump straight into the grit of today's content, which is really, AI. If we were to think about the use of AI image analysis in histopathology on a spectrum from a very basic and obvious level, like the use of AI in triaging cases, to far more transformative possibilities, where do you think AI in pathology is today?
Yeah. And I'm going to start by making a recommendation to the audience. And I give a separate talk that people can find online on AI fluency. So I think it's really important in this emerging field of AI that we all, and I include myself in that, continue to improve our fluency in this new language of AI. And what you'll see in this talk that I give is, I actually advise people not to use the term AI, but instead to use one of the terms beneath AI, like neural network or computer vision, large language model. So refer to the exact application that you're talking about. But to answer your question, so in the field of anatomic pathology, for the most part, we are leveraging a form of AI called deep neural networks. So pretty much all the products in the field right now are these deep neural networks that can help pathologists do what they do as a diagnostic physician. And to directly answer your question, where are we? I actually think we are in the very earliest stages of implementing AI into the field of pathology. I would say to date, over the past 5 years in particular, the field of pathology finally has come around and really shifted their mindset to accept that the field is going digital. So most labs have spent the past years doing that, building the infrastructure. So it's really just over the past 2 to 3 years that people have been thinking about AI and how AI can add value to the practice of pathology. So I think we are very, very early in this whole process.
Yeah, thank you. And to extend that question to the future, then, I mentioned transformative possibilities. Where do you think AI can take us? Where do you think AI can take clinical and research histopathology workflows?
So I would say the low hanging fruit right now. So the applications, the AI applications that people are implementing now are really focused on workflow efficiency. Why is that? That's because every lab that I speak to, their volumes are increasing, the complexity of their work is increasing. Maybe they've merged with another lab, they don't have enough pathologists. So the entire field is really desperate to have anything that will improve efficiency. And they've latched on to AI pathology to do this. So that's why a lot of the applications, including our own at PathAI, are really focused on this efficiency value. And that can take the form of QC automation, biomarker quantification, case assignment, case prioritization. But looking forward, I think that will continue to be important, the workflow.
Mhm.
But what I'm most excited for the future are really two things. One would be developing true medical value applications. And this, I believe, is largely going to come in the form of advanced companion diagnostics that are AI enabled. In fact, PathAI is working with a number of pharma companies to develop just that, AI develop predictive biomarkers. The second thing that I'm really excited about actually goes back to workflow, but more advanced workflow. We are now leveraging the next generation of AI architectures, things like vision language models, agentic AI systems to really transform pathology reporting and also what I call pathology decision support. So, leveraging these tools will enable pathologists to really elevate their contributions to overall medicine, with the goal being providing the patient the most relevant, the most up to date information that will dictate their next steps, therapy wise and patient management wise.
These are such exciting times, aren't they, really? Now, you've mentioned PathAI a few times there, and I suppose my next question is really going to get into that and what, PathAI can offer. But, I think people generally, and I'm going to label this correctly this time rather than using the term AI, but deep neural networks, when people think about and talk about, deep neural networks in pathology, they often jump straight to thinking about diagnosis. But, PathAI seems to be operating across research, clinical trials, image management, biomarker discovery, as you mentioned, and, clinical deployment of AI too. With all that in mind, could you give us a quick picture of what PathAI is doing in the space?
Absolutely. So, for those people who are not familiar with PathAI as a company, we were founded 10 years ago by a pathologist, Dr. Andy Beck, who is our CEO. And it was really Andy's vision from the very beginning that machine learning, neural networks, AI can improve patient outcomes. So, that is our vision statement, to improve patient outcomes with AI powered pathology. And we've stayed very true to that vision over the past 10 years. And what's exciting now in 2026 is that we are actually seeing this vision come true. It was more theoretical and aspirational in the early days, but we're actually developing products that, at least in part, achieve this goal. And just to give you a sense of our strategy as a company, we refer to something that we call the strategic flywheel. And so, what that is, we've identified that there are opportunities to improve patient outcomes, both on the pharma precision medicine side as well as in the clinical space. And the way we go about doing that is we started 10 years ago working with pharma in research. That led to working and deploying AI systems into pharma clinical trials. That led to working with pharma, as I already mentioned, on IVD, in vitro diagnostic medical device tools that likely will be companion diagnostics. And then all of that work then flows as a cycle into the clinical space for our AI Site DX, our image management system customers to adopt and implement in the real world. And then those learnings then can feed back into pharma research. So it really is a virtuous cycle.
It sounds like an excellent feedback loop, which is just what we need right now with all the data that's coming out in different capacities. You mentioned clinical trials there, and I suppose that's the basis for my next question. How are AI tools at the moment being used within the clinical trials environment?
Yeah. So we realized early on that there was a pretty large opportunity to improve clinical trials through the interpretation of biomarkers that are part of those clinical trials, and especially biomarkers that are interpreted by pathologists. So at this point, we are implementing these tools both retrospectively and prospectively. In fact,
Mhm.
this whole line of exploration led us to developing and establishing a service laboratory of sorts, a CRO of sorts, that's a CAP-CLIA GCP, GCLP laboratory in Memphis, Tennessee, where as we speak, we are running multiple phase 2, phase 3 clinical trials deploying digital and AI pathology tools.
So I'm interested to hear your thoughts on variability within clinical trials then, and the impact AI is having there. Pathologists aren't necessarily wrong, but as humans, we introduce different interpretations into trials, don't we? So how big is the problem of variability, I suppose, within clinical trials and drug development?
Right. And this gets to why we've entered this space. So we've seen pathologist variability affecting clinical trials in every example where we've looked. So we think it's a pretty significant problem that AI can address. And I think embedded in your question is where is this variability and how does it affect the clinical trial? And at least at the moment, we're laser focused on two areas, and that's improving the enrollment of the trial and also improving the assessment of the endpoints at the end of the trial. And I think it's pretty clear to everyone. So if you're enrolling the wrong patients on a trial because the read of a biopsy is incorrect, that's detrimental to the trial because your study population is not what you thought it was. And then I think equally important is the endpoint. So this is how a pharma company measures the efficacy of a drug. So if that endpoint is inaccurate, then that jeopardizes meeting the primary endpoint of the trial and the success of the entire drug program. Both are clearly important, and we see opportunities to improve both enrollment and endpoint assessment, actually across multiple disease areas.
Fantastic, thank you. And to stay on the topic of clinical trials, I wanted to ask you about AIM-MASH AI Assist, which I believe it's a tool that PathAI has developed. Could you tell us a little bit more about that? I think it's just received an FDA marking as well.
You're exactly right. So we are very proud that AIM-MASH AI Assist received FDA and EMA qualification in 2005. It might be useful for the audience for me to explain a little bit about this entire space. So I'll start with this whole idea of qualification, which is a little known regulatory pathway that both the FDA and the EMA have. It's part of the biomarker qualification program. So this is separate from the IVD clearance and approval pathways and is really focused to qualify biomarkers for use in clinical trials. We applied for this program for our MASH algorithm called AIM-MASH AI Assist. And I guess I should also explain what MASH is for those people who might not know. MASH stands for metabolic dysfunction associated steatohepatitis, which is the new name for a disease that was called NASH, non-alcoholic steatohepatitis. And maybe another time I can explain why the name was changed. But this is an inflammatory and fibrotic disease of the liver that really is a public health crisis because a quarter of the world's population has MASLD, metabolic dysfunction associated steatotic liver disease, and then a fraction of those patients will go on to have MASH, which can lead to liver transplant, death, cancer. So it really is a significant public health issue.
I guess it's obviously fundamentally important to differentiate these two different patient groups.
Yes, absolutely. And that's what the AIM-MASH tool is really focused on, measuring in clinical trials for MASH drugs, the enrollment and the endpoint assessment biomarkers.
Yeah.
So there are 4 biomarkers that the AIM-MASH algorithm assists pathologists to interpret. So that's what the tool does.
What about the limitations of traditional MASH scoring in clinical trials?
So the way MASH trials were traditionally done and still done today is to include multiple liver biopsies on the trial, at least 2, one at the beginning and one at and after therapy. And then the pathologist would assess 4 biomarkers on those biopsies. And these biomarkers are called ballooning degeneration, macrovesicular steatosis, lobular inflammation, and fibrosis. Each one of them has a scoring system, 0, 1, 2, et cetera. And the issue is that there's a lot of data to show that pathologists are just not reproducible to interpret these biomarkers. So this is the opportunity to improve the situation with AI tools like AIM-MASH AI Assist.
So, for those listeners who aren't as familiar with this as some of us are, could you tell us a bit more about what the tool actually does?
Sure. And I think it's useful because the work we've done with AIM-MASH AI Assist will really serve as a template. And this is an AI assist tool. So we are developing this AI assist tool for MASH, but we intend to develop similar tools for other disease areas. So basically, we trained this algorithm on 100,000 human pathologist annotations from 6,000 MASH clinical trials. And what it does is, as the name implies, it assists the pathologist. So it will actually provide a proposal for each of the 4 biomarkers that I described, along with colored heat maps, so the pathologist can see what the algorithm is actually referring to when it gives its score. And then the pathologist can either agree or disagree with the algorithm. So this is not about automated AI. This is definitely human in the loop AI, or AI assist, as we call it. And we're very proud that this algorithm was published in Nature Medicine twice, once in 2024, and then the clinical validation was published in Nature Medicine in 2025.
Great. Thank you so much, Eric. And sticking on the subject of clinical trials, then I read something to do with PathAI standardizing inflammatory disease endpoints using a new tool as well. I was hoping you could tell us a little bit about that.
Sure. Yeah. So I believe you're referring to our work in inflammatory bowel disease. So, like I was saying when we were talking about MASH, this idea of providing assist tools to pathologists really is disease agnostic. We started in MASH, we've created a tool in inflammatory bowel disease, and the situation is the same. So now in IBD drug clinical trials, GI biopsies are taken and the scoring system is different, but the goal is the same. You want to measure the severity of IBD, and traditionally this is done using one of 3 scoring systems called Gabos, Robarts, and Nancy. And similar to the MASH situation, pathologists are not as reproducible as we would like in measuring these measurements as measures of enrollment and endpoint assessment. So to address this, we've created a separate tool called AIM High UC that outputs the Gabos score and the subgrades, and then that can then be converted if needed to Nancy and Robarts. But the model is almost identical to what we've already discussed with the AIM-MASH AI Assist.
Okay. Fascinating. And thinking about the clinical trial strategy here, how does AIM HIUC and IBD Explore fit into PathAI's broader clinical trial strategy?
Yeah. So the situation here from a regulatory perspective is different than in MASH, where in the IBD field, the current gold standard actually is not tissue diagnosis, it's endoscopy. So for example, colonoscopy. So where the field is moving is to establish histology as a more sensitive endpoint for predicting mucosal healing and endoscopic remission. So our strategy with these products is similar to in MASH, where we really want to improve the ability to measure IBD endpoints and enrollment. And you also asked me about IBD Explore. So our Explore models are really different than the clinical trial models. These are more upstream, if you will. These are discovery models meant to discover new biomarkers in IBD. We also have a Liver Explorer for discovery in liver disease. But these are really for translational scientists and pharma companies to discover the next generation of biomarkers in these disease areas.
Great. Thank you, Eric. And, you've mentioned the kind of capabilities of IBD Explore there, but, you know, I'm, this is a question that comes up a lot with the field changing so much thanks to deep neural networks, or as I referred to it earlier, AI. But, what kinds of new questions can researchers ask that we wouldn't have been able to with conventional pathology review alone.
Yeah, absolutely. So to summarize how human pathologists grade and score things. So when I was in clinical practice and I see, let's say, a GI biopsy, and I'm trying to assess the level of inflammation, humans are only able to very coarsely and subjectively categorize these estimates. And so that's why the current scoring systems have tiers like mild, moderate. So the opportunity for AI is really to apply machine learning and computer vision to truly structure histopathology data. So it really becomes a completely new data element that we can use now to implement into clinical trials, to measure endpoints and enrollment, or even as new predictive biomarkers. So in GI for IBD, examples of this are we can measure the area of inflamed crypts continuously. We can measure the area proportion of goblet cell mucin to the epithelium. So these are parameters that no human pathologist would even attempt to do because it would be so tedious and so time consuming. So that's really the exciting aspect of AI pathology as applies to this area.
Just fascinating, isn't it, how far we've come in such a short amount of time. But yes, next question then. How do you see AI-derived histology endpoints fitting alongside endoscopy, clinical scores, molecular data, and patient-reported outcomes?
Yeah, I get this question a lot, and I see them all as complementary. So, as I said previously, this structured histopathology data is a brand new data element. So, there's a lot of opportunity to think synergistically to look at this new form of data side by side with molecular data, outcome data, other data forms, and actually apply AI to the whole set of data to see if looking at all of these streams of data together could correlate in a way that's incremental to any of the data elements alone as it pertains to patient outcomes or drug response or molecular status. So I think it's a very exciting new development to combine all these data elements together.
Yeah, certainly exciting. And that's the real next step in my point of view anyway. We've got all of these fantastic tools now, but how do we bring all of this data together? Just to go back to inflammatory bowel disease and perhaps use that as an example, do you think tools like this could help redefine what meaningful response looks like in, as I mentioned, inflammatory bowel disease?
I really do. So as I mentioned earlier, the standard now is endoscopy. So I think it's not a far stretch to imagine that histology, actually looking at the cell populations and the dynamic changes in those cell populations pre-post therapy, wouldn't be a more sensitive way to detect mucosal healing and remission in this disease. We, of course, have to prove this with prospective data, but that is what I believe, that histology driven by AI measurement will be the new standard in IBD for measuring endpoints and enrolling patients.